Clarity Clinical Solutions.
A drug that works in a 45-year-old white man may not work the same way in a 70-year-old black woman, a young Hispanic man, or an Asian child. Yet clinical trials have historically been conducted in narrow, homogeneous populations. This is not just unfair, it is dangerous medicine. Who is missing from trials? About 80% of trial participants are white despite being 60% of the U.S. population. Black participants make up 10% of trial participants despite being 13% of the population. Hispanic participants make up only 5% despite being 19%. Asian representation is under 2%. And adults over 65 make up 60% of drug users but only 30% of trial participants. Drugs work differently in different people because of genetics, metabolism, and physiology. Different populations carry different genetic variants that affect drug metabolism. The CYP450 enzyme system metabolizes 70% of drugs and has variants distributed differently across populations. Women metabolize drugs differently than men. 8 of 10 drugs withdrawn from the market between 1997 and 2001 had greater health risks for women, but were not detected because trials enrolled mostly men. BiDil, the first race-specific drug. Approved in 2005 for heart failure in black patients, BiDil reduced mortality by 43% in a trial of 1,050 black patients. Earlier trials in mixed populations had not shown statistically significant benefit. The drug saved lives because researchers bothered to study a black population. Critics argue race is a poor proxy for genetics, but BiDil proved that diverse enrollment is essential. Without it, an effective therapy would have been discarded. Warfarin dosing disparities. Black patients require 20 to 30% higher doses of warfarin, a common blood thinner, than white patients to achieve the same effect. But for decades, one-size-fits-all dosing was used. Variants in the CORC1 and CYP2C9 genes explain the difference. The FDA now includes race-specific dosing information, but only because diverse studies revealed the differences. Women in clinical trials, a history of exclusion. In 1977, the FDA banned women of childbearing potential from early-phase trials. The policy lasted until 1993. For 16 years, drugs were tested almost exclusively on men. Women experience adverse drug reactions at nearly twice the rate of men. The sleeping pill Ambien was approved at a dose too high for women. The FDA only changed the label in 2013, 20 years after approval. Racial and ethnic minorities remain underrepresented. In FDA approval trials between 2015 and 2019, black patients made up only 7% of participants. Many drugs work differently across groups. ACE inhibitors are less effective in black patients. Interferon for hepatitis C is less effective in black patients. When drugs are approved without diverse testing, minority patients receive ineffective or dangerous treatments. Older adults are the missing majority. Adults over 65 consume 40% of prescription drugs but make up only 25 to 35% of trial participants. Aging changes drug handling, kidney function declines, liver metabolism slows. Older adults take multiple medications with drug interactions. A drug tested only in healthy 40-year-olds may be dangerously different in a frail 80-year-old on 8 medications. The FDA now requires diversity. The FDORA Act of 2022 requires sponsors to submit diversity action plans for all Phase 3 trials, specifying enrollment goals by age, ethnicity, sex, and race. The FDA can reject or delay an application if the plan is inadequate or was not followed. This is a dramatic shift from voluntary guidance. The NIH has required inclusion since the 1993 Revitalization Act. But progress has been uneven. NIH-funded trials now enroll equal numbers of men and women, but racial diversity remains poor. Reporting of race data is incomplete in 40% of trials. Mandates alone are not enough, they need enforcement and community engagement. Barriers to diverse participation include mistrust from historical abuses like Tuskegee and Henrietta Lacks. Trial sites are located in white, affluent neighborhoods, 85% of sites are in these areas. Eligibility criteria exclude patients with common comorbidities more prevalent in minority populations. And economic barriers prevent lower-income patients from participating. Solutions that work include community partnerships with health centers and churches, which increase enrollment from underrepresented groups by 2 to 4 times. Decentralized trials using telehealth and mobile nurses reach rural and low-income populations. And broader eligibility criteria that reflect real patients instead of ideal trial participants. Precision medicine needs diversity. 78% of genomic data comes from people of European descent, who represent only 16% of the global population. This means genetic risk scores and drug metabolizing databases are far less accurate for non-Europeans. The NIH All of Us program aims to collect diverse genomic data from 1 million Americans. So here is what we learned. Trials have historically excluded women, minorities, and older adults. Drugs work differently across populations due to genetics and metabolism. BiDil, warfarin, and Ambien prove the point. The FDA now requires diversity action plans. Solutions include community partnerships, decentralized trials, and broader criteria. Precision medicine depends on diverse genomic data. Diversity in trials is not a box to check, it is a scientific necessity.